Bismillahirrahmanirrahim. Dear students, Assalamu alaikum. This is Biology 5090 and today we will study Biotechnology, part 3 of chapter 17. Dears, today we will study the last part of chapter 17, microorganisms and their applications in biotechnology. By the end of this video, you should be able to describe the use of fermenters for large-scale production of antibiotics and single-cell protein. And then you should be able to describe the role of the fungus Penicillium in the production of penicillin, which is an antibiotic. Before getting into the main concepts of the chapter, have you ever seen this type of red grapes? I'm sure you must have eaten these type of green grapes, the yummy one. Dear students, I hope all of you love to eat oranges. But let's see what has happened to these juicy oranges. They have been spoiled by fungi. So this is not a simple fungi. This is fungi Penicillium notatum. The previous photomicrographs which you have seen, which I told you, like the red grapes or the green grapes, they are not the green or the red grapes. Rather, it is the spores of fungi Penicillium, and they have been stained by using different biological dyes. Penicillium was discovered by Sir Alexander Fleming in 1928. Let's study definition of antibiotics. Antibiotics are chemicals produced by certain bacteria and molds, which are also a type of fungi, which are widely used to treat many infectious diseases caused by other bacteria. For example, penicillin, cephalosporins, amoxicillin. These antibiotics may either kill or inhibit the growth of bacteria. The mass production of antibiotics began during World War II with streptomycin and penicillin. Today, most antibiotics are produced by stage fermentations in which strains of microorganisms, either bacteria or fungi, which produce high yields, are grown under optimum conditions of temperature, pressure, in a nutrient media in large fermentation tanks holding several thousand gallons. For many years, scientists knew that certain molds killed certain bacteria. However, researchers needed to understand how to harness this antibacterial microbe and to manufacture enough of the substance before they could make a useful medicine. Now here you can see that the penicillium is growing on oranges, and if we look at the microscopic view of penicillium, okay, these are the spores and this is the thread-like hyphae which we call as mycelium. These basically they produce antibiotic penicillin. So the strains of this penicillin, they are grown into large steel fermentation tanks by adding different kinds of sugars and other ingredients. This process increases the growth of penicillium. When the scientists knew that the penicillin is being formed, they separate it from the mold. Finally, the penicillin is produced, it is purified, and then it is marketed as a medicine. Now let's study the internal structure of a fermenter or a bioreactor. These fermenters or the bioreactors, they are large steel tanks designed to keep the inside environment favorable for the biological process to operate. The main nutrient which is added is lactose along with certain salts. These salts include organic sources like corn steep liquor, and other mineral salts are added. The pH is controlled and is in constant check with the help of pH probe. Since the microorganisms, they divide and multiply for several days inside, so the temperature is also maintained with the help of cooling jacket. The cold water enters from inside and hot water from another pipe is removed, and this way it removes the excess heat from the fermenter. The impeller helps to mix the ingredients, and it moves with the help of motor. From the base, spiragur is present from where sterile air is passed. This sterile air provides oxygen, plus as it moves in the form of bubbles, it also helps in mixing. When the microorganism, they divide rapidly and they grow rapidly and they produce their products, whatever antibiotics they are producing, that is produced after that, it is harvested from the pipe, then they are crystallized and then packed into the bottles and marketed. Single cell protein refers to the total protein extracted from the pure culture of microorganisms like yeast, alga, bacteria. So basically the single cell proteins, which are known as SCPs, they are dried cells of microorganisms which can be used as dietary protein supplement. They are also used as animal feed and can be used for human feed as a protein supplement, also known as novel food and mini food, sometimes as microbial proteins, since we are extracting them from the microorganisms. 60 to 80% dry cell weight contains nucleic acids, fats, carbohydrates, vitamins and minerals. Different strains of fungi, yeast, alga and bacteria are used for the production of single cell proteins. In the large scale production of any type of SCPs, for example mycoprotein or spironula, the fungi is grown in specially designed fermenters like that for the formation of insulin or for penicillium. These fermenters, here the culture medium is used, maybe the glucose, it may be any petroleum byproduct, along with mineral salts, and the temperature is maintained at 30 degrees centigrades. When the SCPs are produced, they are filtered, purified, dried, and then they are marketed as single cell proteins. SCPs normally they have the texture of meat, and they are with blonde taste, so flavoring and cooking can overcome that blonde taste. They provide high amount of proteins. Since most of the world is malnourished, so that is why they are good source of proteins. Also they provide us with vitamins, amino acids, minerals, and since they are rich in fiber, so prevents constipation, provides with instant energy. Plus they are also excellent and convenient source of proteins and other nutrients for feeding the cattle, since we are in need of more meat and milk from the cattle, so they are also a rich source of food for them. Like with any food, SCPs also have certain limitations. Since nucleic acid content is very high, normally 40% if that SCP is formed by alga, 10 to 15% if the SCP is formed from bacteria, and 5 to 10% in the case of yeast. So the high content we get from alga. The presence of carcinogenic and toxic substances may be there. Contamination of pathogenic microorganisms may also be there, indigestion and allergic reactions as well. Production of food grade SCP is quite expensive. So that is why still if a person has to take SCPs, he must consult his physician. Hopefully students, you have understood the topic. Now assignment number 1 is you have to write down the advantages and disadvantages of continuous operation system. Assignment number 2, what is batch operation system? Write down its advantages and disadvantages. Assignment number 3, watch online video regarding discovery of penicillin and write an outline describing how the methods used in penicillin's early production are in contrast to the large-scale commercial production of the drug today. Write your answer for 10 marks and submit me in the next 3 days. Thank you. Dear students, for better understanding, it's advised you to watch the video at least 2 to 3 times. You can also watch certain online videos and lectures regarding the topic. Allah Hafiz, eat healthy and stay safe.